Cell signaling is an essential part of our body. It helps keep our body healthy, stable, and normal. And the G protein cycle is one way cell signaling works. The first image shows us the unactivated G protein coupled receptor, G protein, and enzyme. The ligand or signaling molecule hasn't activated the receptor yet. Therefore, the G protein and enzyme aren't activated either. In the second image, the receptor is activated due to the appearance of the ligand or signaling molecule. Because the receptor is activated, the G protein is in the process of getting activated as well. The guanine diphosphate detaches from the G protein as it kept the G protein inactive, and then the guanine triphosphate attaches to the G protein, which allows for the G protein to be activated. In the third image, the activated G protein has binded and activated the enzyme as well. In the fourth image, the G protein hydrolyzes the guanine triphosphate into guanine diphosphate, and the enzyme in this particular image has also released a phosphate as a cellular response. The activated enzyme relays signals to the other proteins, causing a signaling cascade. Next, we are talking about the tyrosine kinase cycle. The whole chain starts off with a signaling molecule, or ligand. Once the signaling molecules touch each strand of the tyrosine kinase protein, they both move closer together, creating a dimer. Then the dimers release 6 adenine triphosphates, which release 6 adenine diphosphates, which mean the tyrosine kinase proteins are now activated. The fully activated kinase proteins produce phosphates that then draw the inactive relay proteins toward them. The inactive relay proteins attach to the phosphates and become activated. Each of the relay proteins produce a different cellular response. And now we have reached the end of the tyrosine kinase cycle. Serotonin is commonly known as a feel-good transmitter. Serotonin is released and sent to the serotonin transporters. Usually, after this happens, the serotonin is reuptook by the reuptake transporters. The reuptake transporters keep the serotonin levels at bay. However, when ecstasy is taken, the serotonin is morphed and therefore consists of a different compound that can't be taken up by the reuptake transporters. This leads to overcrowding in the synapse cleft and overstimulation in the transport receptors. This leads to a very good feeling for a short time and causes clouded thoughts and intense sensations. After this effect, the serotonin is destroyed and leads to unpleasant feelings similar to a hangover. There are no specific treatments for the intake of ecstasy.